You do?
Circle resistant.
Actually. That's
Cause I have this two current class. Just about Monday nights, so like when
Yeah.
people in the library and just like really distributed and just
That's
like
how.
I kind of picked the worst time to take the group chat.
Did they pick it or do you get to like?
No, she chose it. I think I'll do this.
Yeah, that's an old
Like,
one.
I don't like, you know. At least it's not the fun stuff, you just.
I could, what do they talk about?
Sometimes we do books, or like she happens to like kind of politics.
Should we just?
Advance when it airs.
Yeah, so I think we should.
Yeah.
And we can all just like.
After this meeting.
Yeah. Stick it out.
So you had it last semester too?
Last fall.
It's about.
Well, only freshmen and sophomore year. There's also.
Alright, let's get started today.
Okay, sorry.
Today, you guys on your toes, like super focused, super dialed in. There's gonna be a lot of regulation going on today. Like, that means that there's gonna be stuff that if the system's going fine, it's gonna be one way, if something goes wrong, it's gonna switch and do a different path, right? So this will be exactly like the second question of the motion, which is you need to figure out what's going on, so I can the other big thing I need you guys on your toes about this, today's gonna be a little rough, is that there's gonna be checkpoints occurring all the time, right? Remember for mitosis, we're talking about cell dividing. You can break it down, we're doing it in a new way, right? Like this, where we have, our G1, we have our S phase, and our G2. Right? You guys should be pretty comfortable with these guys, right? G1, the cell's gonna be growing, like that's the main they go. And the S phase is where we're actually talking about DNA. And the G2 phase is basically doing that last little setup of growth to get you to the mitosis part of the disease. And here, remember this is it's broken down even more. PPM, right? And this is followed by solid diseases. This is when mitosis. So we're coming through here, we double up our DNA, we're growing our cell, double the DNA, keep growing the cell. Then we start taking all the little small steps, right? So this was before you condense your DNA, you start removing your nucleus, you start letting it fall apart. You start creating your centrioles, you can grab onto your DNA, you start lining them up, and then you make sure it's everything lined up, and then you can pull. Right? Once you pull, then you can decide, okay, I'm gonna split up, we'll start making a nucleus, make up the DNA, and then we cut the cells, right? This is the thing we talked about last week. Today, I'm gonna bring in all the different rules and places where we have little checkpoints that make sure that is it the right condition to grow, there's gonna be a checkpoint there, right? Across this whole area, we're always gonna have the mechanism in the background saying, is our DNA okay? If the DNA is hurt, there's gonna be places to fix it. The other one is, once you go through here, it's gonna make sure that all the DNA is replicated. When you get up here, it's gonna be that, all these replicated is fine. And then over here, you're gonna have the checkpoints like, do we have the chromosomes lined up so that both sisters can be sure? Right, so we're gonna be jumping around the table. If you're ever feeling lost, that's the thing where I want you guys to go back to your notes later on or go back to the textbook to make sure that wherever we're trying the mechanism, we can understand what part we're putting into this, yeah? So I'm gonna try going through the slides a little bit as we go, so I can emphasize how the mechanism works. And this is where on Monday, you guys are gonna be working and doing the hard thing of when we bring up a mechanism, I want you to then go and figure out where does it fall in here, or in here, or in here, or here, and what it's doing to that process. Yeah. So that's what you guys are gonna have a little bit of fun. Just want this to warm us up, and then we'll start getting into it.
In most cells, Checkpoints in the cell cycle prevent a cell from entering one phase if the events of the preceding phase have not been completed. At the G1 checkpoint, the cell checks for damaged DNA. Arrest at the G1 checkpoint allows repair of the damage to take place before the cell enters S phase, where the damaged DNA would be replicated. The S phase checkpoint provides continual monitoring of the integrity of DNA to ensure that damaged DNA is repaired before it's replicated. In addition, the S phase checkpoint provides a quality control monitor to promote the repair of any errors that occur during DNA replication, such as the incorporation of incorrect bases or incomplete replication of segments of DNA. A DNA damage checkpoint in G2 also leads to cell cycle arrest in response to unreplicated or damaged DNA. This checkpoint prevents the initiation of mitosis until DNA replication is completed. Another checkpoint, called the spindle assembly checkpoint, arrests mitosis if the chromosomes are not properly aligned on the mitotic spindle and therefore not organized for equal distribution to daughter cells. These cell cycle checkpoints function to ensure that complete genomes are transmitted to daughter cells.
Good, I think we're on the same page. All right, so you can see we're gonna be hitting those 4 areas today. And then we're gonna get into exactly how they work.
Sorry, what does it say in blue on the top?
Here we have one on this side, which is gonna be your G2 checkpoint. That's gonna be getting activated by DNA damage, just making sure you have all the copies. In here, like in the mitosis, you're gonna, your big checkpoint here is gonna be the one where we're gonna focus on chromosomes. So this one, I brought it up last time, it was those kinetic cores, right? The centrosome, the kinetic cores pulling it apart.
So there's 4 main checkpoints.
That's this guy. All right, so this is a little bit of background, but the big thing that I want you to take from all of this, or I'll have a super emphasis on this, is, here we go. All this regulation, everything that's gonna be happening here, is gonna be under the control of protein kinases. So this is gonna be the big thing we're gonna be talking about today. And these are gonna be basically our control of cell division. Alright, so what's the kinase, guys? Kinase. Sound, kinase? Is it
Sorry,
kinase, what does it do? It's kind, likes to give stuff away, what's the kinase do? Phosphorylation, right? So one modification here is to keep an eye out. Kinase is going to be the big thing of giving us the phosphorus with the groups. What's its opposite thing? What does the opposite job? Guys remember this one?
phosphorylase.
Okay.
Phosphorylase.
So this is going to be like our positive signal giving us phosphates. The alternate idea is thinking about phosphorus. Say it again.
Phosphorylase.
Phosphates. So this basically removes the phosphates. And you guys can be very comfortable with this. This will be as, because throughout the lecture, you guys are going to be keeping track of how we're modifying phosphates. We're going to be adding them and taking them away, and that's how we're going to turn things on and off. All right. So when they were looking at the background, trying to figure out how stuff works, this is where they started actually picking up some of these kinases that are involved in this pathway. In this case, what they found is like when the cell was dividing, or this one was already dividing, they would steal the cytoplasm of this one, give it into another cell, and you can force it to start going into the division, right? So they were like, hey, there's got to be some signal carried over. This is where they figured out one of these guys. Maturation promoting factor. This was before we knew what it was, so we just gave it this general name. Now we know that in there, what we're going to find is going to be kinases, right? Another example is going to be over here. And here they called it the start checkpoint. It was basically the one that they were looking at in this phase here, this G1 checkpoint, right? So remember, here your cell's growing, get big enough to start DNA. So the big thing is your signal's coming in to get it to progress through this area. In this case, here it's the kinase that they found. It's going to be the CDK1, and the presence of that thing is going to be signaling for it to actually get into the division, make sure it's the appropriate size. This is important because when this thing divides, you can see the daughter cell is not actually of the same size, so it could be a smaller cell, right? And just so that cell doesn't divide even more so and get smaller and smaller over generations, that thing won't divide until it hits enough of that critical signal to be able to be like a big mother cell that lets it go into the pathway. But again, the presence of that molecule is gonna be the big signal of let's let the process go forward. So when we're thinking about the CDKs, we're gonna be thinking about them over time. We're gonna be thinking about, it's gonna be messy today. I'm gonna move this for a second. So focus on the kinases, right? There's gonna be a time component, right? And they're gonna be present at certain time points. Okay. The other thing that's gonna be a big layer is gonna be concentration. So in this case, in order for the kinase to work, it has to be present. The other thing is, depending on how much of the kinase you have, it'll tell us whether it can do a good job or a great job, right? In this case here, you can see that when they started looking at these cyclins, or they found these proteins that seem to change in concentration over time, you see that they kind of build up. So this whole guiding us from that interface into mitosis, right? So we're building up the signal. Even though they're present here, right? Because they're at lower levels, the signal isn't that strong. Here, where they're critical at providing their function or where they're doing the control mechanism, that's when you have these high levels building up. Now it's like present and high concentration, getting the signal to actually work. That kind of makes sense? Yeah. All right, so as we go through today's kinases, like you're gonna see, I'm gonna keep an emphasis on when they're showing up. So now that we've got a little bit closer and we actually started looking into the how, like what's actually there, what's the gene that's there, what does the protein look like? We go figure out that these kinases are actually built up of two different components, right? So this maturation promoting factor, that's the first one that we found from that first example on the left, where they moved the cytoplasm. So for this one, we're breaking them up. Make sure you feel comfortable with both of these. So one half is gonna be our cyclin. So this is the molecule that activates this bottom version, which is your CDK. Your CDK is your cell division kinase. That's the one that actually gets the job done, phosphorylates protein, right? So then, in order to control this, what's gonna happen is the CDKs are always present, they're there the whole time, right? The thing that's gonna be fluctuating over time, the thing that we're gonna have tight control over to regulate cell division is gonna be the presence of the signaling molecule. So for this one, I need you guys to be comfortable with this table, going from when the complex is made and who its partner is, right? So what you're gonna see here, if we go back to our table, there's gonna be in this G1 phase, right? What you guys are gonna observe, this is when the presence of cyclin D is gonna show, right? It's gonna be available, it's gonna start building up as the cell's getting bigger and bigger, giving us that first signals and control here. Its accumulation is gonna end up activating the following signal. This one is responsible to food, growth hormones come in, activate this, right? This is the pathway of how cyclin will activate its own kinase, they'll go to their job, and one of their big roles is they go and they activate the next cyclin in the row, to activate cyclin. So that would be the next one. Now this will be the main molecule that's gonna be regulating from G1 into the S phase. I'm gonna go into details today exactly how this molecule even helps to make sure we only make one copy of each chromosome and that every single chromosome is getting copied. This is a super big play, right? After that, you can see we're gonna jump into our next one, which is gonna be our A cyclins. These are gonna kind of take over from the S phase, so when we're copying DNA. I'm not gonna go into that much detail about that one. And the last one is, at this last phase, you're gonna have a new set of cyclin complex, right? Still be cyclin A, cyclin 1, cyclin 1, but cyclin B is gonna come in. And this is gonna be the big helper in giving us the final signal of we're trying to get the cell to divide, right? So for this one, I need you guys to remember these, where they work, what complex we're thinking about, and also what cyclin is gonna be the main regulator there. My way I remember this is the DEA bros, right? So I know this pathway, I know those G1, G1S, SM, and the DEA bro, DEA bro. That's how I remember it, yeah. So as we're going through our stuff today, we're kind of gonna bring them up here, right? When you're adding them up, I want you guys to be thinking that D is gonna be somewhere up here, building up somewhere in this time point, right? We're gonna set off our cascade, this will end up sitting in the next molecule, which is our E. This one will be kind of taking over in this area and pushing us forward. Then over here, our S molecule is gonna take over, pushing us forward. And then up here is where we're gonna see that, D, E, A pushes us to S. And then our cyclin up here is gonna be the A plus T work together to be the signals when we start getting into the division itself. Yeah? Is that kind of making sense how we're building up the story together? Yeah, alright. So this is gonna be stuff that I'm gonna now go into detail about how they actually work and stuff. Alright, so this is the nicer version of my crude drawing over there, but here we're keeping track of these different cyclic molecules, the different little comments we were talking about, right? So here we're looking at a cell, you're imagining, here's just a regular cell before it's even decided it's G0. It doesn't know it wants to do it. It's gonna get into that point where it's gonna get into the G1 phase to be like, okay, now it's time to start to divide. So there's gonna be growth hormones and there's gonna be food. Those will be the signals that set it off. This is gonna get us to first make, again, that first type of signals, right? You're signaling these. Signaling these are gonna set off this guy. So this will be our next molecule, right? This will be that cyclin E. And this is the one that's gonna be present during that phase of let's make the cell get bigger, get it ready to go into the synthesis. This molecule, I'm gonna show you guys today when we go into how it actually, like I said, sets up, start copying the DNA. That's the job. After that, the next molecule is gonna take over, which is next, guys? Which cyclin? A, yup. This is gonna help us to push through that S and G2 phase. It actually stays on for a pretty long time. And this is just ensuring that we're copying the DNA. And then right at the end, we're gonna see that the last cyclin's taken. This is our B. Our cyclin Bs then are gonna be able to help us. First, I'm gonna show you guys how they initiate mitosis, and then how they basically get us through the passage of actually splitting up the cells. All right, so that's the big mechanism. So for this part, let's do the story one more time. Here I'm actually going into the actual details of what happens, right? So if a cell's in the environment, it's gonna be getting growth factors. Again, this could be hormones from its neighbors, it can be food in the environment. This is gonna turn on these signaling pathways. These pathways are then gonna be responsible for making the synthesis of the cyclin E. So now that we have this in the system, this will be our first signal that, hey, the cell has now decided to go into cell division. And here we're zooming in a little bit more. This is basically the mechanism of how we control it. Here I want you guys to start thinking about, like I said, there's two options as what's gonna happen. Right? The system normally sits like this, where this RB is basically blocking the function of this E2F. So at that point, we're saying one trajectory, nothing's gonna happen, the system's turned off. You have the alternative route, is once the cyclin E shows up, it can phosphorylate the RB, you can get rid of that. The most important thing is it frees up our E2F. That transcription factor can now co-activate genes. In the book, they talk about a lot of different factors. The main one I care about is this. Right? It's the one that leads to the activation of cyclin E. So it kind of cascades the pathway forward. That make sense? All right. So even though it has more targets, for this case, this is the one that I'm going to focus on about today. And up there, the CDK4/6 and cyclin B, that complex, it can signal this molecule. It also goes and targets a lot of different other components. So you guys remember last week, on Wednesday, we talked about how at the end, the stability of the nucleus, how the microtubules work, and what else? And the kinetochores, like they're regulated by kinases that come and signal. These will be the molecules that actually go bind those, do the phosphorylation to turn them off. And we have phosphatases that are normally going to be fighting these little functions. All right, so we get through our E1. We decided to go to cell division. Now we're going to go into how this molecule works. So this molecule, Once it's being made, it normally gets blocked off by this P27. So what you see up there, it's sitting in inactive state. So it can't do its job of doing a kinase function, right? When you start making more of this, you end up creating more of these free units. These units will then go and actually phosphorylate P27s. So what this is gonna end up happening is your population is gonna switch very fast up. If you think about that top part being your off state, they're initially gonna be a lot of those complexes, right? So it'll be the repressor, plus your kinase down here. There's gonna be a lot of them. You're gonna have very few of this one, which will be your active state, just like this. Right? But once you start setting it off, the moment you start, this population starts to go up. Because there's more activators, they'll be destroying more of the repressors. This population starts going down, this goes faster and faster, and this will get brought down more and more. Right? Eventually, you have such a high population of the kinases, you can basically turn this mechanism completely off, and now you're sitting in that super activated state.
So, the cyclin E's that are active were the ones that were just made because of the cyclin D thing. Why were there the complexes to begin with? Did they just not get degraded?
So those, so at the end of today, I'm gonna show you guys that these cyclin, the CDKs and the cyclins, they get regulated at multiple levels. And most of them are working cooperatively to make things happen. So they get regulated at the production rate. They get regulated at, we can modify the kinase function. We can modify them through, like P27 is a protein-protein interaction. So in my head, I think those are like clingy proteins. You can have clingy proteins. We can also modify them with ubiquitin tags to get rid of them, right? So this molecule, these two, this should be building up. There might be some small amount of levels being present, right? So if we go a little more back in time, you can see the problem. This is going to be like, both of these are going to be kind of down like this, right? There's going to be very little. When you start making it, it's going to start trying to build up. But P27 is something that we make ourselves to protect us from cell division. Right? Like its role is like we got to make sure we're not dividing or doing whatever signal is coming out of this. We're not doing that at any other time in our pathway. Right? So this molecule is going to be being present to be like a little safety thing to make sure it's backed up. The moment that we build up enough of the cyclin thing, I can use that. For that to happen is you have to have enough food and nutrients coming in. Let's say we'll be activating cyclin D. Right? When you have enough signal coming in, if the signal goes away, this thing goes away, we stop the signaling pathway. If it's consistently coming in, and this can activate and build up a lot, this one can activate and build up a lot.
Gotcha.
And at that point, we know that we've had a safe time or journey, gives you like a good average time. That would be the powerful thing that now you really want to go forward, right? So it's not like, hey, we had a snack this morning and we're cool. It's like, no, you've been eating well for the last week. It's good for you to go forward. Yeah. But yeah, so this one we're regulation, the P27, I'm gonna bring it up a little bit later because there's a whole family of them, and I'm gonna upload a little table because they're pretty cool. So yeah, P27, that's the cleaning one that blocks the protein's function. Once you activate some of that signal D, it'll start going through that pathway of kicking off its own repressor, leading to eventually it leads to a really sharp window of getting our molecule to show up at really high levels. Right. At this point, one of the really, really big things that this is going to be doing is it's going to mess with this guy. So signaling is going to stop the APCs. So again, this one is something that you want when we're in G0, right? In G0, we don't want to be doing anything involved in our DNA synthesis or copying DNAs or trying to make ourselves double the organelles. So what this protein normally does is the APC has ubiquitin, it tags the cyclins, and it sends them out to destruction, right? So if you're going this way, to G0, where you're staying stand still, the APC is going to kill this, kill A, and it's going to kill the A-B complex, right? So by turning off all those cyclins, when you're like, hey, all those roles or jobs you can carry out are completely turned off. But now, since you're in E phase, and you're like, hey, no, it's time to go into cell division, let's get to work. What it's doing is it's getting rid of the negative regulator. Right, so now our cyclins are gonna be more stable, aka we're gonna be able to build up more. Right, so big task right there. Is this all making sense to you guys? Yeah? I don't know if my drawings are helping or not, but.
How does it stop the APC?
Give me a second. I'm gonna have to look this one up for you. I don't know if it's a protein thing or if it's degradation thing. Keeps exactly working. All right, let me look it up real quick. Cyclin E stops phase promoting complex. I'm gonna have to look it up. I thought it was gonna be a quick one, but I'll look it up and I'll get back to you. Let me get back to this. All right, cool. So you can see we're pushing forward. We've turned off this big negative regulator. So let's go on to the next thing. Sorry. So now, oh, sorry. Yeah. The other big task, another thing that E molecule is going to do. This one is going to be super, super, super important. Right. So at each chromosome, you're going to find this first thing called the origin recognition complex. Right. So let's say you have a chromosome, and this is going to be sitting there like this. Your ORC. Now your goal is you need to copy each chromosome, right? The process is you bring in, you're going to open it up, and you have to bring in this thing called polymerases. They're DNA polymerase. They're DNA dependent DNA polymerases. So grab onto the DNA. What's before single strand is going to get opened up, and as it's going copy into this molecule, it's going to make two chains of double stranded. So it's going to look something like this. Right, you had an initial strand, your polymerases come in, and then you end up with two double-stranded pieces of DNA. Now, the thing that you want to think about here is, how does the cell know that it's made a copy of each chromosome, right? That's going to be a thing where you're like, okay, well, the polymerases can grab on, go to the end, and you're done, right? But how do we know that it's not going to do it again by accident? Grab your old chromosome, the new one you just made, bind to both, and create 4 copies, right? Like, what's the big mechanism? And that's where these guys come in. Again, that ORC is going to recognize the site. From here, I want you guys to focus on the big player up on the left in yellow, that mini chromosome maintenance protein. So this is the protein that's involved in helping open up the DNA ahead of the polymerase. So it's easier for the polymerase to kind of walk across it and make copies of the DNA. This is going to work by first binding to the DNA in this inactive state where it's not phosphorylated. So they'll sit there, it'll kind of grab on. Then what's going to happen is, here's where our cyclin E is going to come in. So here for this one, I want you guys to know cyclin E, and I want you guys to know DDK. They're the ones that are going to signal to give us that phosphorylation event that's going to happen here. Right? When they do this phosphorylation event, now these become an active state, and now this protein can kind of, it'll help shift in front of the polymerase to let it be able to make the copies efficiently. The cool thing there is let me look at this one. Give me 1 second, guys. There's a cool mechanism here. And you have to find it. Yeah. So this case, this molecule, the DDK, like, it's critical in the sense that it's required to make these phosphorylated, so they start working opening this up. If we leave it in their inactivated state, even if they grab on, they won't actually start getting their job done. So the cool thing about this is the E molecule helps activate this process, but the cool thing is that there's a later cyclin that's going to work backwards. Oh, sorry, cyclin D is the other one that gives us information. Cyclin D actually sets this guy up, right? So that means that we need cyclin D signal, but we also need cyclin D signal coming in from this side to activate this. So it means that starting and copying the DNA from the center is gonna happen around this phase. Once we get later in the pathway, even though E is present, since D is not, it's not gonna work. Right, so it'll be a cool way to make sure that when we start copying the DNA once, if you let cyclin D be present even longer, what would happen is you would finish copy your 2 chromosomes, these complexes would land again, and then it would start copying them 2 more times. You end up having 4 copies of each chromosome, right, which would be really bad for you guys. All right, so that's the important one, yeah.
What is CD, or what is the cyclin deactivate?
It, you guys in the drawing, we'll put it as cyclin D drawn into DDK, but it actually goes like this. It's like CDK4 cyclin D, it hits this E2F. This one hits the next guy, which is this D5F4, and this thing turns on to DK. So in my notes, I'm gonna put the arrow that draws directly into it. This is the actual background, right? But for my tests and stuff, the one I really care about is just cyclin D and cyclin E coming in. So in your notes, you can say, I know I put it somewhere here. Yeah, for this, I want you guys to fall in love with the phrase called signaling cascade, right? So there in your notes, I want you guys to draw here, cyclin D activates DDK through a signaling cascade. I'm gonna let you guys get away with that one there. In reality, it's this thing in the background doing the work, and that's your signaling cascade. This molecule turns its transcription factor, it's the next one, this transcription factor finally gets the creation of protein. Yeah.
So what does the DDK do and how is it different than what the cyclin E does in terms of phosphorylating?
I believe the CDK is the one that helps with the phosphorylation, and then you have this other factor, this genes proteins, and help us create the active complex.
Then what does the DDK do?
DDK is the phosphorylation. So phosphorylation coming through this side, this is bringing the genes protein over here.
So, and the GINS protein is just needed to.
To kind of build a complex to work together, right? So it's like, I can't remember exactly what their job is, they help the HEDA case, but it's like, I remember before we could recruit the polymers to the DNA, but it wouldn't do anything to the mediator, kind of grabbed on, got phosphorylated and send it like a way to move it forward. That's what they're basically setting up, right? That phosphorylation event brings the protein complexes. Now this thing as a whole thing working together, now you know. But the cool thing is, again, the big things that we're coming back to is going to be that cyclin E presence and cyclin D presence being over a small window of time relative to each other is going to ensure that we only copy our chromosomes one time, right? So here you guys can imagine if there's errors like cyclin D is going to be there way too long, you're going to start making more copies of your DNA. If the time lapse between E and D doesn't completely overlap, you won't be able to start copying your DNA, you know?
Wait, where is the cyclin D in this? You said that they are both active.
Yeah, this is the one we're gonna do the CDK cyclin D. Just a little arrow going this way. Let's make that purple. And that's the one I was telling you about. Signaling cascade, right?
Oh, I thought that was cyclin E.
No, that's cyclin D works through these 2 transcription factors to make DDK. So that's coming in from one side basically, right? From this side getting us the DDK to work. On the other side, you see the cyclin, CDK2 cyclin E. On the left, the green one. That one's acting as a kinase and it's phosphorylating this, what's it called, recruiter protein or recruiting factor. But by phosphorylating, I think it ends up releasing it, and that's that GINS do its work. So we'll make this a little bit pretty, but I'm gonna go into a little bit more when I get back to the regulation stuff. Is this making sense so far for this part? Yeah? All right, so right now we just went through this first part right here, right? We're like, hey, are we gonna know that we're gonna start copying our DNA? Cycling D and E working at this very early stage, just literally have an exact mechanism to make sure we're going to start copying the chromosome only once, right? That's a big control mechanism we have there. So now we're gonna jump a little bit going forward. We're gonna jump into what's going on with mitosis itself. Right? So here we're gonna get into another cyclin. This time we're jumping a little bit ahead. We're getting all the way to the B1, right? So we're looking at this process up here. So again, this molecule is normally not present, and then it's gonna show up and start building up into the mitosis phase, right? It's gonna go find, work with this partner called CDK1, that'll be the kinase that it partners up with. Now that kinase, when we have a complex, they should be able to try to activate. This is where you're gonna see that there's multiple levels of regulation on top of this. So it's not just about can you make cyclin B. When we get to how the CDK1 can work itself, I want you to take note of, there's 3 phosphorylation markers. This 161, when it gets phosphorylated at this site, it helps it work as a kinase. So that means like, it's an activating one, right? That's gonna turn the gene on. That's gonna be carried out by that CDK activating kinase, CAK, right? So I put here the little plus sign on top and put the little phospho on here. The crazy thing is, if you look at amino acids 14 and 15 there and there, those are again our other 2 places we can have phosphorylating marks. The thing is there, they work the opposite. If they're not there, the system is active. The moment they get phosphorylated, the system actually turns off. Like you end up inhibiting the kinase, right? So that's where WEE1 is going to be the phosphorylates 15 right here, turning the system, turning this kinase off. Even if you have a lot of cyclin B and a lot of CDK, you can have that signal come back and tell the system to stop. And just like I said before, kinases and phosphatases are all over the place. This is where CDC25, it tries to counteract what WEE1 does, right? So if you imagine that if cyclin B CDK kinase is like trying to go into a pathway to make something work, the CAK is a positive signal, pushes it forward to keep going, right? The WEE signal is going to be the negative thing trying to stop it, but you can counteract it with how much CDC25 that you have. Right? So those 2 forces, you can kind of regulate more WEE to stop it, or more CDC25 to let it keep going forward. Then, so imagine when we have a lot of CAK, we have a lot of CD25, our system's functioning, everything's proceeding forward. That's when you have the active things happening. Your cyclin B and the CDK1 are giving us all those phosphorylation marks on all the protein targets we talked about. Right, they should be activating things. Yeah.
What phosphorylates the 14?
They didn't bring that one up in the book. I'd have to look it up for you.
Okay.
I feel like it was I think the book was talking about eukaryotes in that section, but it might be I was mixing them up because they were talking about eukaryotes and yeast back and forth. But I'll look it up for you.
But it's not the WEE1. It's like something different.
No, I think it's something different. Yeah.
But the CDC25 is what dephosphorylates both.
Both of them, yeah.
Okay.
So that WEE1, they bring it up because I believe later on in like the regulation stuff, they bring it back up again.
Okay.
That's why they like super emphasize it. You know?
Okay.
So yeah, so that should be fine. Just feel comfortable with that. CAK is a general activating mechanism, and then the other 2 kind of switch back and forth, competing for each other, right? All right, step 1, step 2, we're getting the job done. So you imagine, going back to that drawing, right? You can activate your system, you have your cyclin B working, and eventually it comes back crashing down, right? Now we're gonna focus on how we get this part of the job done. In this case, what's gonna happen is, what's it called? CDK1 phosphate, APC. Yeah, so in this case, this CDK1, it activates the APC. So remember the one before I told you that anaphase promoting complex is the one that its job is to get rid of all these siblings, right? That's its main role. So this is probably a different kind of phosphorylated mark. This is an activating one. This will end up creating your APC. APC comes back and basically destroys this cyclin. And at that point, it's like, you're done copying your cells, right? You've already split them up. You don't need any of these molecules anymore, and you make sure we clear the system of everything that we have present. So this will be a way that it resets itself to still have the CDKs, but the cyclins themselves are gonna be gone from the system until we decide to go to another cell division, and then we have to reactivate the pathways, yeah? Cool, so. And now, instead of just focusing on one specific window of time, now I'm gonna talk a little bit more broadly of what's happening throughout this whole phase. G1 getting us all the way up here to the G2 checkpoint, right? And that's what we're gonna be keeping an eye out, or your cell's gonna be protecting us against DNA damage. So for this, there's gonna be two types. So you have two strands of DNA, either one breaks or two pieces of DNA break. If two of them break, this ATR is gonna be the first protein that's gonna come in. It recognizes those. If only one, sorry. ATR does one, ATM does if two of the strands break. Yeah. In your papers, when I ask you guys about this one, for the most part, I normally am pretty lazy about these. I always do like DNA breaks. Can't spell it, sorry. Breaks. And then I write them like this, ATR, ATM. I'm a little bit, it's my lazy way of saying like, one of those two pathways is gonna take care of it, because they're both doing kind of the same thing. But if we follow their trajectory, what they're gonna end up doing is, they lead to this phosphorylation of this checkpoints or the CHKs, right? When they do that, they end up inhibiting that CDC25, right? And you guys remember what CDC25 does for us, right? Yeah, no, anybody? CDC25?
Dephosphorylase
That's right.
14 and 15.
Mm-hmm. So right here, so remember that little switch that we had here on these smaller amino acids 14 and 15? Kinases are trying to get it phosphorylated to slow it down. CDC25 is there trying to hold them at bay, saying, no, keep going, keep going. If your DNA is broken, right, you don't want to divide yourself, you want to keep going through the process, it's going to stop you at that point, make you wait there. Cyclin B is still going to be there, CK1 is still going to be there, but it's going to give you time for you to try to copy that DNA or fix that DNA with the other strands. So yeah, so that'll be our big molecule there. On the other side, we're going to see that's going to work with this thing called P53. So P53, when everything's fine, it's normally unphosphorylated and it's consistently getting degraded, right? So if you imagine a little time course for P53, I would be thinking that it's doing something like this, just staying kind of put at a low level. When the system gets activated, ATR, ATM, you're gonna have your signaling cascade. It's gonna lead to the phosphorylation of P53. That's gonna make it more stable. It's gonna build up. It's gonna go find this, one of the targets is gonna be P21. Now P21 is gonna be one of the molecules that they inhibit the cyclin, CDK cyclin complexes, right? These are through protein-protein interactions. In my head, I'm almost thinking like they're like a cleaning thing, like you just grab on it. They'll stop our signaling, right? So if the DNA damage is actually pretty small, it stops down, it slows down those cyclins for a bit, right? Giving us a little bit of a time to like, hey, let's hold up, let's make things, let's try to fix things. If the DNA damage cannot be repaired, there's too much going on in different spots, you can build up more and more and higher levels of the P53 to the point where this is the molecule that'll tell your cells to kind of commit apoptosis, like to kill themselves, right? And that's the way that your body normally keeps track of, like, if the cell has too much DNA damage, we can just completely shred the cell, right? But if you guys remember, it'll open up. At that point, all of our other cells will kind of just go around eating it. It'll shred all the DNA, shred all the proteins, and it'll be like a hard reset. But when this thing gets mutated or broken, what's gonna happen is we're not able to put the brakes on cell division. And that means your cells will try to keep going forward, they'll divide, and just keep accumulating more and more DNA damage, right? This one's really important when they bring up cancer, because it also has signals from, like they get into P53 that come from when cells don't have enough oxygen, or if there's not enough nutrients in the environment. And that's why like tumors and cancers, even though you're not supporting them that well with blood flow, they still keep dividing because they don't have that ability to stop themselves, right? And they keep going. Also, they can work in like, what's it called? Environments with low oxygen. They're actually like thriving. but it's because they don't have race to synchronize. All right, does this one make sense? Yeah? All right. Let me see what's gonna play with you guys. So for this one, I want you guys to kind of think of these cyclin CDK complexes as like partners, but I want you guys to think of them as being general. So let's, like, for a second, imagine it didn't say cyclin B, it just was cyclin, it was A, right? D, E, A, or B. The things that I want you guys to think about is how are we gonna do the big regulation of these guys, right? On one side, we have control of the cyclins by when we make, we talked about the D one, it depends on the food. The E cyclin depends on being present. Can't remember when we brought up the D one, but making them at a specific time gets regulated. The other places you can regulate them as they're being made, it's gonna be these phosphorylation marks, right? So 161, we can kinase that function at that spot, we can activate the signal. At the other side, 14 and 15, that's where we have our switch that we talked about, right? We can phosphorylate, turn it off, CDC25 can come back and turn it back on. The other molecule that I don't have here, but I kind of would want to draw in somewhere over here is going to be the one I was talking about, the clingy proteins. So for these, in the book, they're put up as a general category, and they're called CDK inhibitors. So like this, CDK inhibitors. There's a little table with them on the book. I didn't have a chance to put it, I couldn't find it through the images to put it in today, but these are pretty cool molecules, right? So for these, I want you guys to be thinking about, they normally call them in two groups. There's gonna be the INC 4, and there's gonna be the CYP, KIP, would be the other family. So these are basically two groups. When you go into them, you'll see that there'll be a list of these, which will say like P21, P27, what are the other Ps? P21, 27 are the ones I have right off my mind right now. But like P21, right, so I think it's in this group, or P21, oh, sorry. These two groups, the main thing here is INC4 is going to do a lot of signaling when it comes to the G1. So on that first checkpoint, the CYP kit is going to be doing a little bit of info on both. It's going to follow us when we do our G1. It's also going to follow us when we do the SG2 phase. These molecules, their whole job is they're normally activated, or their role is to put a pause on the system, right? Because what they do is this activated complex, once everything's together, they'll come to that complex and bind to it and block its function for a while. So they're a way to kind of slow things down if something's going wrong. Yeah. They're not as drastic as the other ones where, like CDNF protein complex, this actually means their protein getting completely destroyed. And at that point, we lost the signal that we built up obviously to produce it. These guys just kind of hold it at bay for a little bit. Once they're done or anything is fixed, they can let go of the complex and the signal's still technically present. Yeah. All right, so.
So is the P53 a CKI, one of the inhibitors, or is it different?
P53. So P53
Because
works
I
a
think
little
it
bit.
said like
This
CDK inhibitor.
this guy, right?
Mhm.
So P53, it itself is a transcription factor. So once it gets phosphorylated, if something's going wrong, it'll go grab onto that piece of DNA and it makes P21, right? And then P21, I hope I'm not saying this wrong. If you're getting rid of cyclin E, all right, let's figure this out with you guys. Cyclin E, what phase are we in?
Yes.
S or what are the possibility?
G1.
Answer G1. Okay. It's probably gonna, it could be in either one, I think, but it's probably gonna be here. So let's say, we'll double check it in the book, but P21 will be one of the molecules right here. This one is gonna help us stop cyclin E. Like that'll be its job. It'll go hold on to it. Once the signal tells it that it's okay to keep going forward, it lets go. Now this guy can get back to work. Yeah. All right. So we have, let me go back to my pathway. Okay, so we said production of cyclin, phosphorylation of cyclin, kinase phosphatase spikes to regulate cyclin. We have the CDK inhibitors coming in right here that can hold on to stuff. And the final thing is, this is the final regulation, which is the APC, right? The anaphase promoting complex is, we're officially done with cell division. That one's only gonna be working with cyclin B, as in cyclin B activates it, and at that point, you're gonna clear the system of the other cyclins, right? And the goal there is, we just finished going through this whole pathway, we've doubled up our DNA, we split it apart, and now our next phase is we're technically jumping back to G0. And in G0, we don't want these to be present at all. So that's what this is gonna super activate this system to make sure that we're good, like we're out of this programming to try to make the cell divide. Yeah? All right. Give me 1 second, guys. Put it down. But P27 was another one that I liked from down here. So P27, again, one of those proteins that its job is to block one of the cyclins, right? Just hold it at bay, set it not to work. The cool thing about that one is that P27, it starts out here, at G0, G1, and we kind of always have it present. So there's that protein P27 is always doing its job and telling your cell to stay in G0 and not do anything. Only when the signals come in, the food, the growth hormones, they hit that signaling cascade. One of the things they go turn off is P27. By turning off P27, you basically relieve your cyclin Ds, right? So that's gonna be that first signal up here. Getting rid of P27 is gonna let this guy do its job. So you can build up that first part of the signal, right? So at the same time, you're gonna bump up cyclin D levels, and you're gonna get away from the mechanism that only protects it. I'm gonna give you guys a little bit of questions about cancer or how stuff could work. There, the only thing you guys need to think about for Monday's class is gonna be, you guys doing that worksheet, you guys working with each other, or you work on your own, I'll send it to you guys, but here I show you guys how there's power, how they regulate, right? Some of the things you can do to make it go forward, some things you can do to take it off. When you think about mutations or stuff getting broken, in some of the cases it'll be like, imagine I make it so that 3 sites can never get phosphorylated, right? What would happen there? That means that if you made the weed protein trying to stop this pathway, it wouldn't be able to do it, so no matter what we were trying to tell what's wrong with the environment, it will go forward. Same thing with your break P27, right? Cells are not gonna let it. So the P27 goes up, you'll proceed forward. So I'll give you guys puzzles like that, and you guys tell me what you think would happen. So there's gonna be positive factors go bad and you're not helping anymore, and you're not gonna do the divide, or negative factors are broken and things are go crazy and not doing it.
So if P27 was broken, that's when like the little cells would continue to divide and grow.
Yeah, because they wouldn't, they'd be able to build up the cyclin D, they can like, there's enough food to keep going.
Okay.
Cool, alright. I have a talk on Monday, I'll send you guys an email if you guys wanna go to it, it's Monday at 4:30, and then, yeah, alright, good luck, I'll give it to you.